terms of current research, there are a couple of studies going on to look at better medications to cut down on the rate of aneurysm, even after IVIG treatment. As mentioned, about 5% of patients, even after IVIG, still develop aneurysm, and we would like to get that down to close to zero as possible. So there are some studies looking at other or adjunct therapies. Then there have been some relatively recent studies and interesting studies in mice looking at whether specific medication could prevent this remodeling of the vessels. So as I kind of demonstrated, this kind of process where you have the eventual sometimes occlusion of the blood vessel, there are medicines that can slow down this process if needed. But the difficulty with this question is sometimes you do need a little bit of remodeling to recreate a vessel. The question is how much should you get, and how can you, in those patients, slow down that process? So in mice, there have been some studies. And then finally, just to talk about my own research, was funded quite a long time now. I had a one-year grant from Vasculitis Foundation, and it was looking at a particular protein called follistatin-like 1 protein in Kawasaki disease. And so this was the initial study where we found that patients with aneurysms in Kawasaki disease had higher levels of this particular protein than patients with Kawasaki disease in the middle there who did not have aneurysms. And so the question was, what does this protein do in Kawasaki disease? Now, this is a protein that everybody has in their blood, and there's no, as far as we know, there aren't people who have, none of this, because it's a very, very important protein. So you can't be born without this protein. So really, it's thinking more about what is this gene? What does this gene do? Is it something that could be blocked, cause a problem, or is it helpful? And so this is a cross-section of a mouth part, which just shows on the right a normal mouth part, and the red is the lining of the heart. The red here is the lining which shows where the protein is expressed. And on the left is a cross-section of a heart, and you can see all this extra red is the extra epithelial-1 that is expressed all over the heart. So it's not only in the blood that we see epithelial-1 after Kawasaki disease, but it's also expressed all over in the heart. And so when I looked at trying to block this protein, what we saw that was without this protein, mice who got a very high dose of an agent that causes them to develop a Kawasaki-like syndrome seem to have a decreased rate of survival. So it seemed to suggest protein was protective. And when we also looked at the hearts of these mice, we found too that mice who have this protein which is knocked out have higher levels of inflammation. So all of these graphs, the leftmost bar graph, all these points on the left of these three graphs show the increased number of blood cells in the knockout mice as opposed to the normal mice. And so it's really telling us that this is a protective gene, a protective protein. When we did something very high tech, we took these little cross sections of the heart, and we actually dissolved them almost into cell suspension. So sort of every single one of these dots represents a little cell that we kind of dissolved and made in a kind of almost like a liquid form. And then we could tell which cell was which using a sophisticated genetic technique. And we then wanted to see what would happen, what is happening in our mice that don't have these genes turned on. And what we found is in many of the cells, and this is our gene that's knocked out, we should see that, and this is a very, very complicated chart, but basically what it tells us is there may be a gene, another gene, which has got a fancy name, EBTB16, that is related to FSCL1, and that without FSCL1, you get increases of these two inflammatory genes. And so it's telling us a little bit about the mechanism behind FSCL1. So there'll be next steps. But so this study was, initially something that had been a long set of work, and initially the initial results were not as interesting as they are now. But that's where we are today with the research.